proteins. Okay, so which one is very important? Is the DNA or the transcription or the protein? Which one is very important to study actually? Is one is better than the other?
The protein.
Why?
Because DNA has a lot of junk and the protein is the actual product.
Okay, well, so protein doesn't have a junk at all, or what?
I mean, I guess there could be some redundancy, but
Redundancy of what?
Like multiple, like DNA making multiple of the same kind of protein.
Okay, so okay, let me ask you. Okay, so you just said DNA is junk, so protein is good. Yeah, okay, that's fair. Okay, but do you really think we should exclude DNA altogether?
Not altogether, but if you wanted to like compare based on function or like phenotype, it makes more sense to study the protein.
Okay, what about epigenetics?
Then DNA is more important.
Okay, so epigenetics, DNA is very important. What about transcripts? What about mRNA?
And yeah, then transcription is important.
Okay, because you know gene regulations gene regulations are very important. Am I right? Do you think without gene regulation, protein functions by itself? Yeah. So regulation is very important. Yeah. So I think it's very important to study everything, you know. But the point is that having a protein, a lot of things that you can answer, like the way you said the phenotype. Yes, that's true that we can answer a lot of questions in protein level, but that doesn't necessarily mean that DNA is bad. So, for example, one place where we can study more DNA more is actually gene editing. Yeah. Do you think that gene editing can be done in a protein level?
No.
Yeah. So where can we do the gene editing?
In the DNA level.
Because then you can alter the missense, am I right? Alter the frame shift mutation, or alter the end product, am I right? So how the proteins are being synthesized, am I right? So basically, you know, the DNA is the genetic code is very, very important, you know, for anything, you know, you do. It's very, very fundamentally very important. Of course, there's a lot of you know, errors are there in the genetic codes, but that's fine. It's part of life, you know. So, but studying protein is very, very important, you know. But same as, you know, it's very equivalent to studying DNA. Okay, a lot of things, you know, you can study in protein that you may not be able to study in DNA, but vice versa, you know. So they all are very important. Anyway, so the structure of proteins, we'll talk about some of the structure of the proteins. So basically proteins usually, you know, they have a very important, crucial biological function, you know, they can fulfill, you know, and most of the structural proteins are three-dimensional conformation. They have a proper folding. Okay, so why misfolding is bad? So what is proper folding?
You just get the right configuration of the protein.
Okay,
of the sequence.
Yeah, that's fine. So, but what is misfolding? Is misfolding is bad?
Can misfolding lead to like diseases?
That's right. So misfolding leads to diseases. Okay, it can cause aggregations. So proper folding is always very important. So do you know which ones are actually facilitated with the proper folding? What look, you're just muted. Just mumbling yourself.
I don't know if it's right, but my brain thought Golgi apparatus.
That's okay. That's okay. Golgi apparatus. Where is the Golgi apparatus coming from?
In the cell?
Huh? You mean the Golgi apparatus is involved in the proper folding? You mean?
That's what I think, but it might be wrong.
Why would you think that?
Because I know when proteins come after they're transcribed,
Okay.
it goes through the Golgi, and then the Golgi like modifies the protein before it exits the cell.
Where did you get that information from?
Am I saying the wrong organelle?
Well, Golgis are very important for trafficking. Yeah. So for sorting functions. Yeah. So sorting functions, like, but not necessarily in the context of misfolding. It's only for sorting. So, for example, endocytic pathways, or so ER Golgi are very important in the trafficking of a lot of vacuolar and mediated proteins. Okay, but in terms of the proper folding, can you think about which ones are very important for that facilitates in the proper folding? Proper
mean
folding.
Like
Huh?
chaperonins?
Yeah, chaperones. Yeah, that's a good point. So chaperones. So chaperones or chaperonins, and those are the ones that maintain the protein homeostasis. So chaperones play a major role or facilitate in the proper folding of the protein. Okay, so very important protein family. Okay, so there are a lot of chaperones. So can you just throw in some examples of some chaperones? Think about it. Just take a guess. So what type of chaperones are there? So what happens when the protein is actually, if you heat the protein?
The protein will denature.
So did you guys do any lab work with the denaturation? You should have. Did you guys run any SDS-PH gel? No SDS-PH gel?
I don't remember anything like that.
Did Mark showed you how to run an SDS-PH gel? Like electrophoresis.
Oh yeah. All those labs were online, so I don't remember them that well.
Yeah, so those are SDS-PH gels. So those SDS is a sodium diacetyl sulfate, pH, mean like electrophoresis, so it's a pulse electrical field, pH, pulse agarose, PAGE, gel electrophoresis. Yeah, so basically when you have a protein, the protein denatures. So I think probably in this biochemistry class, I think Mark should be able to show you how to run a Western blotting or SDS-PH gel. So usually when you run an SDS-PH gel to separate the proteins, you usually denature the protein, because the denaturation will allow you, you heat the protein. Okay, so but these proteins, but there are some proteins from chaperone family that can sustain heating. Okay, so that there's no denaturation. So denaturation can lead to there won't be any proper folding. So meaning that's why you can separate the protein by heating them. Okay, so chaperones. So what type of chaperones are there? That can help us facilitate their proper folding? So heat shock proteins. Yeah, HSPs. Yeah, HSP 10, HSP 60, HSP 40. Yeah, HSP 30. A lot of heat shock proteins are there. Okay, these all are chaperones. Okay, all right. So now, the proteins, they're very important for three-dimensional conformation. They're important for fulfilling a specific biological function. The most important thing is they have a proper folding, and the native fold will have a large number of favorable interactions within the protein. So of course, the most important thing is there's a conformational entropy of folding is there. Okay, to form an native fold. Okay, so there are 4 different levels of proteins structures that we are going to study. Okay, so those are the primary structure, the secondary structure, tertiary structure, and quaternary structure. So the primary structure consists of a sequence of amino acids that are linked together by peptide bonds. Okay, and usually they contain the disulfide bonds. Okay, the secondary structure is usually an helix, an alpha helix. Okay, and meaning the polypeptides are arranged in in a unit, what they call the alpha helix units. Okay, and the helix is part of a tertiary structure of the folded polypeptide. You see here, that's a tertiary structure. Okay, and which itself has got, if you have all these assembled subunits, that makes up quaternary structure, and this is what we call them as a macromolecular complexes, meaning multiple subunits forming a complex. Okay, so for example, let's say here, okay, so this is one subunit. Okay, this is two subunits. Okay, this is three. Okay, okay, and this is called macromolecular complexes, meaning assembly of many different subunits to form a macromolecular complexes or macromolecular assemblies. You guys follow? Okay, all right, so there are 4 different levels of protein structures out there. Okay, so now the structure of the protein is partially dictated by the peptide bonds. Very, very important, peptide bonds. Okay, so I'll give you one example. Okay, so what's an open reading frame? What's an open reading frame?
It's just genetic material until you hit a stop codon.
Okay, so it's a series of amino acids. Yeah.
Or I guess, yeah, series of amino acids that are coded
Okay,
for.
and yeah, and then you have a stop codon, the start codon, and these codes are already usually in the triplets. Yeah. So now the question is, when you have a huge protein, and what are peptides then? So proteins are nothing but a series of amino acids. Yeah. And what about peptides? What are peptides?
There are chains of those amino acids.
So it's a very short chain of amino acids. Yeah. And how do you cut the how do you make a short chain of amino acids? So how do you break the protein into peptide?
With proteases.
Good. So you can use the proteases, for example. So you can use trypsin. Yeah. So trypsin is one protease. Okay. You can use it. You can use lysine. L-lysine. Okay. So there are a lot of proteases out there. Trypsin is one where it specifically cuts into arginine and lysine. So this particular specific protease goes and cuts into arginine and lysine. And now, and wherever you see an arginine and lysine, it goes and cuts it, and that forms a short amino acid. And short amino acids chain, which is called a peptide. Okay, so in this case, the protein structure is usually dictated by peptide bond. We'll talk about this peptide bond. The peptide bonds, the resonance cause the peptide bonds. Okay, and usually they are the resonance are very quite rigid and planar. Okay, and they're very less reactive compared to the esters. Okay, and they exhibit a large dipole moment. We'll talk about all those things, in the subsequent slides. A large dipole moment. Okay, so the resonance are very important for causing the peptide bonds. Okay, and the peptide bonds dictate the structure of the protein. Okay, so the definition of resonance here, the peptide bond is as follows. All right, so here basically each peptide bond has got a double bond character due to resonance, because they usually are very rigid and they cannot rotate. Okay. Because the bond between the carbonyl carbon and the nitrogen has a partial double bond character, rotation around this bond is very restricted. Okay, so that's why the peptide bonds are usually, peptide units are usually in the planar side. They're very rigid. Okay, and because of that, the rotation of the peptide bond backbone is very much restricted. Okay, so in this case here, the carbonyl oxygen has a partial negative charge. The nitrogen amide, the amide nitrogen has a partial positive charge. So the positive and negative will form an electric dipole. Okay, and all peptides, okay, all peptides will occur in transconfiguration. Okay, so, and that's resonance. Okay, so peptide bonds form the structure of the protein, and the resonance are the one that cause the peptide bonds. And the peptide bonds are typically have a carbonyl oxygen that are partially negative charge, and an amide nitrogen that are partially positive charge, and that forms a dipole. Okay. All right. So now we'll talk about secondary structures. Okay. So the secondary structures refers to a spatial arrangement of the polypeptide backbone. There are two regular arrangements are there. One is alpha helix, another one is a beta sheet. So alpha helix are usually they are stabilized by hydrogen bonds between nearby residues. The beta sheets are nothing but they are stabilized by hydrogen bonds between adjacent segments. Okay, and they need not have to be nearby. And the random coil is nothing but an irregular arrangement of a polypeptide chain. Like for example, you can imagine, you know, there's no straight path, you know, it's just all tumbled up, you know, it's all coiled, you know, so that's why it's called random coil. It's an irregular arrangement of a polypeptide chain. Okay, so we'll just talk about each one of them. Okay, so basically a random coil is nothing but it's a polymer conformation where the monomer subunits are oriented randomly while still being bonded to adjacent units. Okay. Okay, the alpha helix. The alpha helix has an helical bone that is held together by hydrogen bonds between the backbone amides. Okay, and usually the alpha helix is like a right-handed helix, something like this. See here, this is how the alpha helix looks like. It's a right-handed helix. Okay, and peptide bonds are aligned roughly parallel with the helical axis. Okay, so basically the alpha helix, most important thing here, it's the helical backbone is held together by hydrogen bonds. And they are typically they are right-handed helix. Okay, and side chains point out or roughly perpendicular to the helical axis. So this is how it looks like. Okay, the alpha helix. Okay, so the alpha helix also has the inner diameter of the alpha helix is usually 4 to 5 angstroms. The outer diameter of the helix is about 10 to 12 angstroms. Okay, they happens to fit well in the major groove of the double-strand DNA. Okay, and the residues align very well, nicely on top of each other. Okay, so these are general properties of the alpha helix. Okay. Okay, so here are some of the models. We'll talk about it. Okay, so I think this is very important. Okay, so first, this is the ball and stick model. Okay, this one is a ball and stick model. This is the alpha helical model. And this is a space filling model. And this is a helical wheel projection model. Okay, so in the ball and stick model, did I post this lecture as well in the in the PowerPoint slide? Yeah, okay. So in the ball and stick model, they usually show the interchain hydrogen bonds. Okay, and they have a single turn. Okay, they have only a single turn. So that's about 3.6 residues. Okay, that's not that. It's about with 5.4 angstroms to 3.6 residues. So basically, they are being. This model shows the interchain hydrogen bonds, and they have only single helical turns. Whereas the case of alpha helix model, Okay, basically, if you look at the longitudinal side, you'll see some sort of a purple, some spears here. Okay, so this actually emphasize, a halo structure here, because it's another way of viewing your alpha helix structure. Okay, so there's no fixed rule, like which model you want to use. You can use either way, either one of the model, just to gain more information about what each models can offer you. Okay, so but in the case of the space filling model here, again, that shows the alpha helix are in very close contact. Okay, whereas the case of helical wheel projection model, you can see that here, each one of them, each number represents some sort of a hydrophobic structures, okay, of the polypeptide. Okay, so the red and the blue residues illustrate the negative and positive charge side chains of the residues. Okay, so basically each of the surfaces, okay, are being colored here. Okay, so the yellow ones are all the hydrophobic structures. Okay, again, when you have a structure, we'll talk about how we can crystallize the structure and everything by using NMR and X-ray crystallography. And once you have a structure, then we can use any of this representation or representative models to visualize your structural information. That's the whole point here. Okay, there are different ways you can actually view it. Okay, the structural information. Okay, so sequences affects helical stability. Okay, so what are the sequences that affects the helical stability? Well, we have a helix formers and helix breakers. Okay, so what are helix breakers? Proline and glycine are the helix breakers. Okay, so when you have a proline and glycine, they can break the helix. But what forms the helix? Alanine and leucine are the one that forms the helix. Okay, so, and those are very, very important. Okay, so this helical formers and helical breakers. Okay, so helix dipole. Okay, the helix dipole are nothing but the peptide bond has a strong dipole moment, that is a carbonyl O negative and amide H positive. Okay, so all peptide bonds will have the similar orientation. The alpha helix has a large macroscopic dipole moment. Okay, so the dipole moment are usually the carbonyl O negative and amide H positive. Okay, so the negatively charged residues often occur near the positive end to form a helix dipole. Okay, so that's what the helix dipole means. Okay, so it's basically the positive or negative charged residues. Okay, so here's an example here. How the helix dipole looks. This is an electrical dipole of a peptide bond on an alpha helical segment, and you have all this positive, negative charged molecules of the amino and the carbon and the amino and carbon constituents of each peptide are indicated by plus and minus symbols. Okay, so the amino group and the carbonyl group of each peptide bond are indicated by plus and minus. And now you can have a charged molecules, and you can have an electrical dipole because you have a positive, negative constituents of a peptide bond. Okay, so the non-hydrogen bonds, bonded amino acid and amino and carbonyl constituents of the peptide bonds, are shown in this red color. Okay, now we'll talk about the beta sheets. Okay, talked about the alpha helix, so now we'll talk about the beta sheets. So here in this beta sheets, the peptide bonds, the peptide bonds are arranged the beta sheets are sheet-like arrangement of a backbone that are held together by hydrogen bonds, okay, between the backbone amides in different strands. Okay, so something like this here. Okay, this is a beta strand, and you can see that how the backbones are held together by hydrogen bonds here. Okay, so between this amide groups, there's always the hydrogen bonds are holding this amide backbones. Okay, here in this case, they form a pleated sheet-like structure, the beta sheets. Okay. So there are two different types of beta sheets are there here. So parallel and anti-parallel beta sheets. Okay. So in the case of parallel beta sheets, the H bonded strands run in the same direction. Whereas the anti-parallel ones, the H bonded strands run in opposite direction. Okay. So usually the anti-parallel are much more stronger in terms of the H bonds, whereas in the case of parallel beta sheets, the H bonds are a bit weaker. So here is your anti-parallel ones, and then we'll talk. So here in this case, you can see here how the beta conformation of the polypeptide chains are arranged here. So basically, what it is showing is you can have the hydrogen bonds here between the adjacent chains. Okay, so just to remind you guys, so just anti-parallel means beta sheets H bonded strands run in opposite directions, and parallel beta sheets means H bonded strands run in the same direction. Okay, so here are the ones that are in opposite direction. You can see how the hydrogen bonds are actually running. Okay, so and okay, so whereas in the case of here, so here's the anti-parallel beta sheets where the hydrogen bonds are being shown in the opposite directions here. Okay, you can see here all the hydrogen bonds are in the opposite directions. Okay, because the hydrogen bonding pattern is different here. Whereas in the case of parallel sheets, if you look at it, here the hydrogen bond cross links between adjacent chains. Okay, and you can see here again, going back here, the parallel means here the beta sheets of the H bonded strands run in the same direction, and here is where the parallel beta sheet, they runs in the same direction they run. They're not in the opposite direction here. Okay, so here are the parallel ones where you can see they are in the hydrogen bonds are pretty much in the same direction. Okay, so not in the opposite direction there. So this is a parallel sheet. So you have two different sheets there. So anti-parallel and parallel sheets. So parallel sheets where H bonded strands run in the same direction. And anti-parallel, the H bonded strands run in the opposite directions. Okay, so we'll talk about beta turns. Okay, so the beta turns are typically occur whenever the strands in the beta sheets change the direction. Okay, there's about a 180-degree turn is there, which can be accomplished over 4 amino acids. So the proline in position 2 or the glycine in position 3 are the most common ones in beta turns. Okay, so this is how the beta turns change the directions. Okay, so now here's one. There are two different types of beta turns: type 1, type 2 beta turns. Type 1 turns occurs more twice as frequently than the beta type 2 turns. Okay, and usually the type 2 turns are usually occur in the glycine as a third residue. Okay, that's where the type 2 beta turn occurs. Okay, and yeah, okay, so and this is how the structure of the beta turns looks like. So beta 1 and beta 2 turns. The beta 2 is most commonly occurring in the glycine, third residue of the glycine. Okay, now we'll talk about how we can measure this actually, the secondary structure of the proteins. So we can measure the secondary structure of the proteins using this UV circular dichorism. Okay, so it's using an ultraviolet circular dichorism. Okay, so usually the circular dichorism can allow us to study the secondary structure of the protein. They help in examining the folding and binding properties of the proteins. Okay, they are routinely used in the pharmaceutical companies. Okay, and also people who are interested in studying the protein conformation and stability. Okay, so the circular dichorism is nothing but it's a delta E, so it's an entropy where you can have a molar absorption difference of light of the left and the right circular polarized light. So basically, you can measure the absorption on the left side or the right side of the. If you have a prism, you can just see how much of the light is being absorbed on the left and right side direction, and then you can calculate the entropy actually of the absorbed light. Okay, so the chromophores in the chiral environment produce a characteristic signal. CD signals from peptide bonds depend on the chain conformation. Okay, so basically the bottom line is in this entropy. So you're basically calculating the absorption difference on the left and right of the circularly polarized light. Okay, so for example, here in this case, this is a circular dichroism spectroscopy where you have an alpha helix, beta conformation, random coil. So random coil, we know that there's no structure, there's no proper structure, so it's all over the place. So that's what you're seeing here. There's no structure here. So that's a random coil here. Okay. And here you can see a different wavelength. You are measuring how much of delta E is there. Okay. So this is a CD spectrum that's giving you a rough estimate of the fraction of the protein made up of the secondary structures. Okay, so and usually you are measuring here how much of, after several different wavelengths, you are measuring how much denaturation or the conformational changes are happening in the structure in solution. Okay, so and you can measure based on this CD spectrum. Okay, so circular dichroism is very, very important to study the secondary structures, and you can actually even figure out, how stable your conformational structures are, in terms of alpha helix, beta conformation, and random coil. Okay, and here in this particular specific slide, we are measuring the delta E, which is based on the left and the right side of the absorption of the absorbed light. You are measuring the delta E here. Okay. Okay, so next we'll talk about protein tertiary structure. So tertiary structure refers to the overall spatial arrangement of atoms in a protein. So usually they are stabilized by numerous weak interactions. They're largely hydrophobic and polar interactions. And as we know, they can be stabilized by disulfide bonds. Okay. So, and there are two major classes of protein tertiary structures are there. One is the fibrous and globular. Okay, so we'll talk about what those fibrous and globular structures are. Okay, so here is one example to show the structure of the collagen. Okay, so for example, the panel A shows the collagen structure, alpha chain structure. This is a repeating secondary structure unique to this protein. Okay. So here, what is a repeating peptide sequence? So it's basically, the glycine X proline or glycine X4. We need to see what is this HYP stands for. Just a second. So basically, the glycine and proline are the ones. Okay, I think it's hydroxyproline, I believe. I just want to double check it. I think it's hydroxyproline. So it should be hydroxyproline. I can tell you, in the break, I'll tell you what it is. I'm pretty sure it's hydroxyproline. So, but it can be either hydroxyproline or it can be glycine X proline. Okay. All right. So what's happening here? So now, you have this alpha chain of the collagen, the second structure of the protein, where you have the repeating a tripeptide sequence of glycine X proline or glycine X hydroxyproline. Okay, and whereas in the case of the space filling model here, okay, so this is a space. This is another space filling model where you can see the structure of the collagen. Whereas in the case of this, the helical ones, okay, here you can just see 3 of these helices, helices, okay, basically the gray ones, the blue ones, and the purple ones, okay. All right, so, you know, and these are wrapped around one another like in a right-hand twist, okay. So that's how so we talked about the right-hand twist, you know, how the helices are actually twisted, am I right? So and the final D is actually the collagen superhelical structure. It's like it's represented in a ball and stick representation, where you have a glycine residues are shown in the red color, okay, and here we're just basically seeing, you know, how this superhelixes are arranged. You can just see a little bit of a central, you know, so how this different collagen structures are arranged in a ball and stick, you know. So the red ones are the glycine ones, okay. So this is all these things are just, you know, giving you an example. These examples are just showing, you know, what are different models you can use to represent the structure of the collagen, okay. So each model has its own unique properties anyway, okay. So there's no such thing called, you know, what is the best representation. Whichever representation, it gives you more information, and that's the one that you'll choose to you know, understand the structure in a better way. Okay, so here is the water-soluble globular protein. This is one of the example of the tertiary structure of the sperm whale myoglobulin. Okay, so here the heme group, you know, the red ones are the heme group here. Okay, so basically this is actually showing how the polypeptides are actually shown in a ribbon-like structure. So this is a contour. This is a contour. And this is a ribbon-like structure. Okay, so and basically this is a surface contour image showing the binding pockets. You know, basically where do they bind? So for example, if you take a drug, so assume the red ones here, the heme ones are the drug, let's say. So if you have a protein structure, and if you do a chemical coupling, so what do they say? What they say is actually called chemical soaking, so, or crystal soaking. Do you know what is crystal soaking is? So crystal soaking is actually basically you crystallize the structure of a protein. So, you know, so how do you crystallize the structure of a protein? How do you do that? So you purify the protein. Yeah, you purify the protein and you crystallize them into small crystals. And these crystals, depending on, you know, whether it's a membrane protein or non-membrane protein, because the more you have the alpha helix and alpha helices, the tougher it gets to crystallize the protein. So, but once you have the crystals, and you can take those crystals and use the beam, and you know where the beam is? The X-ray beam. It's not everywhere these X-ray beams are available. So only in specific places it's available. Do you know where it is? In Canada. There's only one place it is there. So all the researchers who are studying crystallography or the structural biology work, people who are studying the NMR, or people who are not NMR, people who wants to do X-ray diffraction, you know, X-ray crystallography,